Scientists at MIT made a discovery in 2018 that most doctors still haven't fully processed. They took aging intestinal stem cells, the kind that repair your gut lining every single week of your life, and after just 24 hours of fasting, those cells doubled their regenerative capacity. Not improved, not slightly better, doubled. The lead researcher, Dr. Ömer Yilmaz, described it as the most dramatic metabolic shift his lab had ever documented in stem cell biology. Then, in 2024, researchers at Columbia University published a paper in Nature showing that a simple cycle of fasting and refeeding could make old blood stem cells behave almost indistinguishably from young ones. And just months after that, the same MIT lab published a new study identifying a single amino acid, one you're almost certainly not thinking about, that activates an entirely separate pathway to expand your intestinal stem cell pool. 3 institutions, 3 separate mechanisms, all pointing to the same conclusion. What you eat, when you eat it, and whether you're getting specific nutrients directly determines how well your stem cells function after 50. Dr. Becker here. I spend a significant portion of every week reviewing primary research literature so I can translate it for people who want the science without the spin. What I'm sharing today is not supplement marketing dressed up as biology. These are peer-reviewed studies from some of the most rigorous labs in the world, and I want to walk you through exactly what they found, what it means for your body, and what you can realistically do about it starting this week. Before we go further, if evidence-based health information is something you value, subscribing to this channel takes about 2 seconds and helps me keep doing this work. No pressure, just genuine appreciation if you do. Here is what we're covering today. I'm going to break down 7 studies, including research from MIT, Columbia, USC, and a 2016 clinical trial that measured stem cell mobilization directly in adults between 50 and 75 years old. I'll explain 5 distinct biological mechanisms, each backed by different evidence, that together explain why stem cell function declines with age and what specifically interrupts that decline. Then I'm giving you a practical protocol organized by priority, not by what sounds most impressive, but by what has the strongest evidence in humans. And I'm going to be honest with you about where the science is solid, where it's promising but still mostly in animal models, and where certain popular claims simply don't hold up. The stem cell conversation has been hijacked by a lot of noise. Expensive IV infusions, unproven supplements with celebrity endorsements, clinics charging $30,000 for procedures with almost no rigorous human trial data. That's not what this is. This is the actual biology, the actual research, and the actual things that the evidence supports. Let's start with the research. The most widely cited study in this space came out of MIT's Koch Institute in 2018 and was published in Cell Stem Cell, one of the most selective journals in regenerative biology. The lead author was Mihailova and colleagues from Yilmaz's lab. They were studying intestinal stem cells, the cells responsible for regenerating the lining of your small intestine. Your intestinal lining replaces itself approximately every 5 days. Every single week for your entire life, stem cells in the crypts of your gut are dividing and producing new cells to maintain that barrier. What Yilmaz's team wanted to know was whether aging changed that process and whether nutrition could modify it. What they found was extraordinary. After 24 hours of fasting in mice, and critically in both young and old mice, intestinal stem cells showed a dramatic increase in regenerative capacity. To measure this, they extracted stem cells and grew them into what are called organoids, essentially tiny miniature intestines in a dish. The fasted stem cells produced organoids at roughly twice the rate of the control group. When they dug into the molecular mechanism, they found that fasting forced these cells to switch their primary fuel source from glucose to fatty acids, and that metabolic switch activated a family of transcription factors called PPRs, peroxisome proliferator activated receptors, which in turn upregulated genes responsible for self-renewal. Here is the important nuance the headlines missed. This effect was present in old mice as well as young ones. That is the finding that matters for this audience. The capacity to upregulate stem cell function via this metabolic pathway does not disappear with age. It just requires the right trigger. The limitation, which I want to be transparent about, is that this study was in mice. The organoid work is compelling, and the mechanism is biologically plausible in humans, but we do not yet have a controlled human trial that directly measured intestinal stem cell regenerative capacity before and after a 24-hour fast. The mechanistic data is strong. The direct human evidence on this specific endpoint is still pending. Now hold that mechanism in your mind, the glucose to fatty acid switch, because it connects to everything else I'm going to tell you. The second landmark study came from Columbia University in May 2024 and was published in Nature. The principal investigator was Emmanuel Passaguet, who runs Columbia's Stem Cell Initiative, and the lead author was Della Russo and colleagues. They were working with hematopoietic stem cells, HSCs, which are the blood-forming stem cells that live in your bone marrow and generate every type of blood cell you have. Red cells, white cells, platelets, every single one starts as an HSC. The central question of the study was, why do HSCs function worse as we age? The answer they found was not primarily about the cells themselves becoming irreparably damaged. It was about the environment those cells were living in. Chronic low-grade inflammation, what researchers now call inflammaging, a portmanteau of inflammation and aging, was degrading the metabolic machinery inside HSCs. Specifically, inflamed bone marrow microenvironments were impairing the cell's ability to perform glycolysis, the normal process of converting glucose to energy. HSCs that could compensate by activating autophagy, the cellular self-cleaning mechanism, survived and maintained function. Those that couldn't deteriorated. The critical finding for our purposes was this: when Passaguet's team subjected old mice to cycles of fasting followed by refeeding, the HSCs were pushed to activate autophagy, and after that refeeding cycle, those aged HSCs performed in transplantation assays almost as efficiently as HSCs from young mice. The phrase used in the paper was nearly as efficient, not identical, but substantially rejuvenated. Dr. Passaguet made a statement in the press release that I think is worth noting carefully. She said, and I'm paraphrasing from her public comments, that the equivalent fasting period in humans would likely be considerably longer than what was tested in mice, and that the practical protocols remain to be worked out. She also emphasized that the real target is the inflammatory environment, the fire in the bone marrow, not just the fasting itself. Reducing inflammaging is what creates conditions in which HSCs can maintain and repair themselves. That is a subtle but important distinction. The fasting was the tool. Reducing inflammation was the goal. The third study I want to cover comes from Walter Longo's lab at USC, published in Cell Stem Cell in 2014. Longo's team was investigating what happens to the immune system during prolonged fasting. Not the 16 to 18-hour intermittent fasting most people have heard of, but extended fasts of 48 to 72 hours. In mouse models, they found that these longer fasts suppressed a signaling molecule called PKA, protein kinase A, and simultaneously reduced circulating levels of IGF-1, insulin-like growth factor 1. When PKA was suppressed, hematopoietic stem cells entered a profoundly regenerative state, essentially clearing out aged and damaged immune cells and then regenerating new ones from HSC populations. This study is particularly significant because it included preliminary data from a human clinical trial, an IRB-approved phase 1 study in cancer patients undergoing chemotherapy. In those patients, periodic fasting of around 72 hours was associated with preservation of white blood cell counts and what appeared to be protection of HSC function during the chemotherapy cycles. I want to be honest about the limitations here. These were cancer patients under chemotherapy, not healthy older adults pursuing preventive health. The results are suggestive and biologically consistent, but they cannot be directly extrapolated to a healthy 50-year-old doing intermittent fasting. What the Longo work establishes clearly, with strong mechanistic evidence, is that IGF-1 and PKA act as a kind of abundant signal that keeps stem cells in maintenance mode rather than regenerative mode. When those signals quiet down, through fasting, through reducing protein intake from animal sources, or potentially through other means, HSCs have the opportunity to shift into repair mode. That mechanism is well supported across multiple model systems. The fourth study shifts to a different type of stem cell and to direct human evidence, which is where the data becomes most actionable. This was a clinical trial published in the Journal of Applied Physiology in 2016, conducted by Montgomery and colleagues, and it was explicitly designed to study adults in the 50 to 75-year age range. The specific cells they were measuring were endothelial progenitor cells, or EPCs, the stem cells responsible for repairing and regenerating the lining of your blood vessels. The study included 45 participants divided into three groups: adults with normal glucose tolerance, adults with impaired glucose tolerance, what we'd call prediabetes, and adults with established type 2 diabetes. All participants completed a single session of submaximal aerobic exercise, 30 minutes on a treadmill at roughly 60 to 65% of maximal oxygen uptake. Before and after the exercise session, blood samples were drawn and analyzed by flow cytometry to count circulating EPCs. In the group with normal glucose tolerance, a single exercise session produced a 23% increase in circulating EPCs. That was a statistically significant result with a p-value below 0.1. In the prediabetes and type 2 diabetes groups, this response was substantially blunted. The mechanism involves shear stress. The mechanical force of blood flowing against vessel walls during exercise activates an enzyme called eNOS, endothelial nitric oxide synthase, which produces nitric oxide and signals the bone marrow to release EPCs into circulation. What this tells us is two things. First, exercise works in humans, specifically in the 50-plus age group, to mobilize stem cells responsible for vascular repair. That is direct human evidence, and it's the most actionable finding in this entire body of research. Second, metabolic health, specifically glucose metabolism, is a prerequisite for that mechanism to function properly. High blood sugar interferes with the exercise EPC pathway. If glucose metabolism is impaired, you cannot fully benefit from the vascular stem cell response to exercise, even if you're exercising consistently. The metabolic environment comes first. The fifth pillar of evidence comes from a 2022 meta-analysis published in the European Journal of Applied Physiology, conducted by Ferentinos and colleagues. They pooled data from 31 studies involving 747 participants across age groups from 19 to 76. Their analysis confirmed the exercise EPC relationship across multiple study designs and quantified the effect size. For aerobic exercise alone, the standardized mean difference was 1.40 with a 95% confidence interval ranging from 1.0 to 180. For combined aerobic and resistance training in cardiovascular patients, the effect size was 1.84. For high-intensity interval protocols using 4-minute intervals, the EPC response was superior to continuous moderate-intensity exercise in sedentary adults. Effect sizes above 0.8 are considered large in clinical research. An effect size of 1.4 is substantial. This is not a marginal benefit. Exercise produces a robust, reproducible, dose-dependent increase in circulating stem cells that repair your vascular system, and it does so through a mechanism that is well characterized at the molecular level. The sixth study I want to bring in comes from the same MIT lab, Yilmaz's group, but was published in Nature in late 2025. The lead author was Chi and colleagues, and the work was focused on amino acids and intestinal stem cells. The team screened all 20 standard amino acids to determine which, if any, had a direct effect on intestinal stem cell activity. The results were striking. Cysteine, a sulfur-containing amino acid found in eggs, chicken, legumes, and cruciferous vegetables, including broccoli, produced the most dramatic effect on ISC expansion of any single nutrient tested. The mechanism involves cysteine being converted to CoA, which activates CD8 T cells in the intestinal tissue, which then release a cytokine called IL-22, which signals intestinal stem cells to expand their pool. This is a completely distinct pathway from the PPAR mechanism activated by fasting. It's an immune-mediated stem cell activation that can theoretically occur even in the fed state, as long as cysteine is available. The limitation is that this study was conducted in mice, and the dose-response relationship in humans has not been formally characterized. But the mechanistic pathway, cysteine, CoA, CD8, IL-22, ISC expansion, is coherent and biologically testable, and it points toward protein quality, not just protein quantity, as relevant to stem cell function. The seventh piece of evidence I want to discuss is a comprehensive review paper published in 2022 in PMC that synthesized the nutritional research across multiple stem cell types. Among the relevant findings, a ketogenic diet sustained for 4 weeks increased intestinal stem cell number and improved regeneration following intestinal injury in mouse models. The mechanism appeared to involve beta-hydroxybutyrate, the primary ketone body, activating stem cells through a pathway distinct from either the PPAR switch or the cysteine IL-22 route. Beta-hydroxybutyrate, for context, is produced during fasting, during low-carbohydrate eating, or during ketogenic dietary protocols. It acts as both a fuel source and a signaling molecule, influencing gene expression through HDAC inhibition. Taken together, these 7 pieces of research describe a system in which stem cell function, across multiple tissue types, is exquisitely sensitive to nutritional and metabolic inputs, not supplements, not injections. The fundamental metabolic signals your body generates depending on how you eat, when you eat, and how you move. Now let me walk you through the 5 core mechanisms, because understanding the biology is what allows you to make decisions that are actually grounded in the science rather than in marketing. The first mechanism I call the metabolic switch. When your body is continuously supplied with glucose, which is the default state for most adults eating a typical Western diet 3 times a day plus snacks, stem cells use glucose as their primary energy source. That's metabolically efficient in the short term, but it doesn't activate the regenerative programs that require fatty acid oxidation. Think of it this way: your body has a primary mode and a repair mode. In the primary mode, it's focused on using what's available right now. In repair mode, it starts optimizing systems for resilience. The switch between those modes requires a shift in fuel source when glucose availability drops, through fasting or through significant carbohydrate restriction. Stem cells shift to oxidizing fatty acids, and that shift activates PPAR transcription factors that upregulate self-renewal genes. The cells become more capable of regenerating damaged tissue. In mice, that switch measurably doubled regenerative output. The biological machinery for this switch exists in humans. The research on the precise human dose, how long, how many hours, how many times per week, is still being worked out. The second mechanism is autophagy activation. Autophagy is the cellular process by which cells break down and recycle their damaged components. Think of it as the cell's internal housekeeping system. In a well-fed, metabolically active environment, autophagy is relatively suppressed. The cell is focused on production, not cleanup. During fasting or caloric restriction, autophagy is upregulated dramatically. For stem cells, this matters because chronic inflammation generates molecular damage within stem cells over time. Oxidized proteins, dysfunctional mitochondria, damaged DNA repair machinery, all of these accumulate with age. Stem cells that can activate autophagy and clear this damage maintain function. Those that cannot drift into senescence or produce daughter cells with impaired function. The Columbia 2024 study made this the centerpiece of their findings. HSCs that activated autophagy during the fasting cycle were the ones that rejuvenated. The intervention was the trigger, autophagy was the mechanism of rescue. The third mechanism is IGF-1 and PKA suppression, which I think of as turning off the abundance signal. IGF-1 is a growth factor that circulates at higher levels when protein intake is high and caloric intake is sufficient. It signals to the body that resources are plentiful and that growth and maintenance should be prioritized over deep repair. When IGF-1 is chronically elevated, which is common in adults eating high amounts of animal protein 3 times daily, it keeps stem cells in a kind of suppressed standby mode rather than in active repair mode. When IGF-1 drops, which happens during fasting and to a lesser extent during periods of reduced animal protein intake, PKA activity decreases, and HSCs shift toward the regenerative program documented by Longo's lab. This is one reason why the research on extended fasting, not just 16 to 18-hour intermittent fasting, but periodic longer fasts, shows more dramatic effects on immune stem cell populations. The longer fast creates a more sustained drop in IGF-1, which gives the regenerative program more time to operate. The fourth mechanism is nitric oxide-mediated EPC mobilization, which is the pathway through which exercise works. During aerobic exercise at moderate to vigorous intensity, cardiac output increases substantially, and blood flow velocity through peripheral vessels increases. That increased velocity generates what's called shear stress, a mechanical force against the inner lining of vessel walls. Shear stress is detected by sensors in endothelial cells, which respond by activating ENOS, endothelial nitric oxide synthase. ENOS produces nitric oxide, which has two relevant effects. It dilates blood vessels in the short term, and it signals the bone marrow to mobilize endothelial progenitor cells into circulation. Those EPCs travel to areas of vascular damage, atherosclerotic plaques, micro-injuries in arterial walls, endothelial dysfunction sites, and participate in repair. The reason this mechanism is disrupted in people with prediabetes and type 2 diabetes is that high glucose concentrations in the blood impair ENOS function through a process involving glycosylation and oxidative stress. High blood sugar is directly chemically hostile to the ENOS pathway. This is not a minor impairment. It essentially eliminates the 23% EPC mobilization effect seen in healthy adults, which is one reason that metabolic health, specifically glycemic control, has to be understood as a prerequisite for this mechanism to work. The fifth mechanism involves what are called WNENT and Notch signaling pathways, which are the molecular switchboards that determine whether a stem cell self-renews or differentiates. Think of these pathways as the navigation system that tells a stem cell what to become and when to divide. In young tissue, these signals are balanced. Enough WNT signaling to maintain the stem cell pool, enough Notch signaling to drive appropriate differentiation. With aging, the microenvironment shifts. Inflammatory cytokines like IL-6 and TNF-alpha alter the local signal landscape. The result is that muscle stem cells, called satellite cells, begin differentiating toward fibroblasts rather than new muscle fibers. Blood stem cells produce skewed populations with altered ratios of lymphoid and myeloid cells. The tissue repair capacity drifts. Certain dietary compounds, sulforaphane from broccoli, resveratrol, curcumin, have documented effects on these signaling pathways in both in vitro and in vivo models. The practical significance of food-derived concentrations in humans is still being quantified, but the mechanistic direction is clear and consistently supported across independent research groups. Now let's talk about what this research actually tells you to do. And I want to organize this carefully because the evidence is not equally strong across all the interventions, and I think it does you a disservice to present them as if they are. The highest priority, with the most direct human evidence, specifically in adults over 50, is aerobic exercise at moderate to vigorous intensity. The Montgomery 2016 trial, the Ferrentinos 2022 meta-analysis of 747 participants, and the well-characterized ENOS mechanism together constitute the strongest body of evidence in this entire space for a practical, accessible intervention. 30 to 45 minutes at an intensity where you're breathing harder than normal but could still speak in short sentences, done 3 to 5 times per week, produces a measurable and reproducible increase in circulating endothelial progenitor cells. In the meta-analysis, the effect size for aerobic exercise was 1.40. For combined aerobic and resistance training, it was 1.84. The data for high-intensity interval training using longer intervals, specifically 4-minute effort periods rather than the short bursts some people associate with HEAT, shows the strongest acute response in sedentary adults. If you are not currently exercising with sufficient frequency and intensity, this is the single most evidence-supported action you can take for stem cell function. It costs nothing. It's available immediately. And the dose-response relationship across the research literature is clear enough to act on. The second priority is managing your metabolic environment, specifically blood glucose and chronic inflammation, because the data is unambiguous that both directly undermine the effectiveness of everything else on this list. The Montgomery trial showed that the 23% EPC increase from exercise was essentially eliminated in adults with impaired glucose tolerance. The Columbia 2024 work identified inflammaging as the primary cause of HSC deterioration. These are not separate problems with separate solutions. They are the same problem from different angles. A metabolically inflamed environment is hostile to stem cell function across multiple tissue types. The dietary approach with the most consistent evidence for reducing both glycemic dysregulation and chronic inflammation is the Mediterranean dietary pattern. Emphasis on olive oil, fatty fish, leafy greens, legumes, nuts, and modest amounts of animal protein, with substantial reduction in refined carbohydrates and ultra-processed foods. This is not novel advice, but the mechanism connecting it to stem cell biology is newer and gives it a more specific rationale. Third priority, ensure adequate intake of the nutrients with documented roles in stem cell signaling pathways. The most evidence supported in this context are the following. Protein quality, specifically cysteine-containing foods, based on the Yilmaz Lab's 2025 Nature study. Cysteine is found in eggs, chicken, fish, legumes, and cruciferous vegetables. The mechanism, cysteine converting to CoA and activating the IL-22 pathway for ISC expansion, is preclinical, but it's specific enough and the foods involved are benign enough that increasing them is a reasonable low-risk action. Distributing protein intake across meals, rather than concentrating it in 1 or 2 large servings, has independent evidence supporting muscle protein synthesis efficiency, and it's compatible with the stem cell rationale. Cruciferous vegetables, specifically broccoli and broccoli sprouts, for their sulforaphane content. Sulforaphane activates NRF2, a master regulator of antioxidant response, and has documented effects on WNT beta-catenin signaling and NFKB inflammatory pathways. Half a cup of raw broccoli sprouts delivers a substantially higher dose of glucoraphanin, the sulforaphane precursor, than an equivalent weight of mature broccoli. The enzyme myrosinase, which converts glucoraphanin to sulforaphane, is destroyed by heat. Raw or lightly steamed, with a brief crush before cooking if you choose to cook it, preserves more activity. 2 to 3 servings of cruciferous vegetables per week is a reasonable target based on the epidemiological and mechanistic literature. Green tea, specifically matcha or high-quality loose-leaf green tea, for its EGCG content. EGCG, epigallocatechin galatase, has documented effects on HDAC inhibition, which influences gene expression in stem cell populations, and has shown EPC mobilization potential in diabetic rat models. 2 to 3 cups daily is consistent with the consumption levels associated with benefits in observational studies. Avoid drinking it with milk, which chelates polyphenols and reduces absorption. Between meals is preferred over with meals for the same reason. Omega-3 fatty acids from fatty fish, salmon, sardines, mackerel, herring. The mechanism here is anti-inflammatory. EPA and DHA reduce circulating IL-6 and TNF-alpha, 2 of the cytokines most directly implicated in bone marrow inflammaging from the Columbia 2024 work. 2 to 3 servings per week from fatty fish sources or a supplement providing 2 to 4 grams of combined EPA and DHA daily is consistent with the research-supported range. The omega-3 evidence for reducing systemic inflammation is among the most replicated in nutritional science. Curcumin from turmeric, consumed with black pepper, and a fat source to enhance absorption. Piperine, the compound in black pepper, increases curcumin bioavailability by approximately 2,000%. A meaningful enhancement that matters because curcumin has very poor baseline absorption. The evidence base for curcumin includes phase 1 clinical trials showing safety and biological activity in colorectal cancer stem cell contexts, as well as extensive in vitro and in vivo work on WNT, Notch, and hedgehog pathway modulation. The food-derived dose from regular culinary use is at the lower end of what produces measurable effects in research, but regular consumption as part of an anti-inflammatory dietary pattern is both plausible and practical. Fourth priority, consider time-restricted eating or intermittent fasting if your metabolic health profile is appropriate. I want to be specific about this recommendation because it is the most commonly oversimplified in popular health content. The mechanism for stem cell activation through fasting, the PPAR switch, the autophagy upregulation, the IGF-1 suppression, is well characterized and credible. But the human evidence for these specific endpoints remains limited compared to the exercise and inflammation data. The Longo 2014 work included preliminary human data, but in a cancer patient population under active chemotherapy. The MIT studies were in mouse models. What we have with high confidence is that fasting produces metabolic changes that are directionally consistent with the stem cell research in animal models. What we do not yet have is a randomized controlled trial in healthy humans over 50 that directly measured stem cell regenerative capacity as a primary outcome before and after a structured fasting protocol. That said, the indirect evidence, metabolic improvements, inflammation reduction, autophagy markers, is sufficient to consider time-restricted eating as a reasonable practice for adults without the contraindications I'm about to describe. A 16 to 18-hour fasting window, done 3 to 4 days per week rather than every day, is a reasonable starting point based on the balance of evidence. If you are currently taking insulin or sulfonylurea medications for diabetes, fasting poses a real risk of hypoglycemia and should not be initiated without direct coordination with your prescribing physician. If you have a history of eating disorders, fasting protocols of any kind are contraindicated. If your BMI is below 18.5, caloric restriction is inappropriate. Before I discuss the timeline of what you can expect, I want to address the supplement landscape directly. There is an entire industry built around what are marketed as stem cell activators. These products typically contain combinations of polyphenols, adaptogens, and proprietary blends with impressive names and citations to cell culture studies. I am not aware of any product in this category that has demonstrated in a blinded, controlled human trial that it produced a measurable increase in stem cell number, function, or regenerative output in older adults. The individual components, sulforaphane, EGCG, resveratrol, have evidence at the mechanistic level, but that evidence is largely preclinical. The specific products using these components have not been validated as complete formulations. The compounds that have the most credible evidence, and I want to name these specifically because I get questions about them, are vitamin D3, where deficiency is highly prevalent in adults over 50 and directly associated with immune stem cell dysfunction, omega-3 fatty acids, for the inflammaging rationale discussed above, and NAD+ precursors, such as NMN or NR, which have small human trials showing biological activity and align with the mitochondrial function story in aged HSCs. These are not magic. They are metabolic support for processes that primarily depend on the lifestyle factors we've already discussed. Now, what can you realistically expect and when? The first thing I want to say is that framing this as double your stem cells in 30 days, while compelling as a headline, misrepresents how this biology actually works. Here is what the evidence does support across realistic timeframes. Within the first 1 to 2 weeks of beginning consistent aerobic exercise, assuming you are metabolically healthy, you will be mobilizing EPCs within each exercise session. That effect is acute. It happens during and immediately after each bout of exercise. The cumulative impact on vascular health and endothelial function begins to accumulate over weeks. You are not going to feel this directly. You will not have a subjective sense of stem cells working differently. What you may notice is improved exercise recovery, which reflects better tissue repair broadly, and potentially improved energy levels, reflecting better mitochondrial efficiency. Within the first month of consistent adherence to the anti-inflammatory dietary pattern with the specific foods I described, you can reasonably expect measurable reductions in high-sensitivity C-reactive protein if your baseline was elevated. hsCRP is the best available proxy biomarker for the kind of systemic inflammation that the Columbia research identified as the primary enemy of bone marrow HSC function. If you want to actually track this, ask your doctor for a hsCRP alongside your standard metabolic panel. The target is below 1 milligram per liter. Many adults over 50 are at 2, 3, or higher. At 2 to 3 months of consistent practice, sustained aerobic exercise, anti-inflammatory diet, adequate sleep, managed glycemia, the research supports expecting improvements in HbA1c if it was in the pre-diabetic range, improvements in fasting glucose, improvements in lipid profiles, and measurable changes in endothelial function measurable by a test called flow-mediated dilation. These are not stem cell counts. We cannot measure your intestinal stem cell regenerative capacity or your HSC autophagic efficiency with a standard blood test. But they are the downstream functional outcomes that the stem cell biology ultimately produces. there are a few things from the research that I think deserve specific mention as practical insights. Sleep is not optional in this biology. Research has shown that sleep deprivation reduces the migratory capacity of stem cells substantially, and chronically elevated cortisol, which disrupts sleep and is produced during prolonged stress, creates the exact kind of glucocorticoid-rich bone marrow environment that the Columbia work identified as damaging to HSC function. 7 to 8 hours of sleep in a consistent schedule is not a wellness nicety. It is a direct input into the inflammatory tone of your bone marrow. Temperature regulation during sleep, keeping the bedroom cool around 16 to 19 degrees Celsius, significantly improves sleep architecture and has downstream effects on metabolic markers relevant to this entire discussion. Glycemic variability matters more than fasting glucose alone. Even if your fasting glucose is normal, large spikes in blood sugar after meals generate oxidative stress and inflammatory signaling that accumulate over time. Eating in an order, fiber and protein before carbohydrates in a meal, consistently reduces postprandial glucose spikes. A tablespoon of apple cider vinegar in water before a carbohydrate-rich meal has modest but reproducible effects on glycemic response through a mechanism involving delayed gastric emptying and improved insulin sensitivity. Neither of these interventions will transform your metabolic health alone, but they reduce the moment-to-moment inflammatory burden that ages your HSC niche. One final note on the science that I think is important to say directly. The research on stem cells and diet is genuinely exciting and it is moving quickly, but it is also a field that has been consistently ahead of the clinical translation. The mouse data is compelling. The mechanistic models are sophisticated. The human clinical trials that directly measure stem cell outcomes as primary endpoints are still relatively sparse. What I've described today represents the strongest available evidence for this topic. It does not represent certainty. The relationship between what you eat and how your stem cells function is real, supported by peer-reviewed data from credible institutions, and worth taking seriously. But any specific claim about a particular food doubling any biological quantity in humans over a defined time frame should be held with appropriate skepticism, including and especially when that claim is made in the title of a YouTube video. What the evidence does support, clearly, repeatedly, across independent research groups, is that exercise mobilizes vascular stem cells directly in older humans. That chronic inflammation is the primary environmental threat to bone marrow stem cell function with age, that the metabolic switch created by fasting or reduced carbohydrate availability activates stem cell regenerative programs in ways that appear highly conserved across species, that specific nutrients, cysteine, sulforaphane, EGCG, omega-3s, interact with stem cell signaling pathways through mechanisms that are specific and testable, and that the combination of these factors, sustained over months rather than 30 days, produces the downstream metabolic and vascular health improvements that the stem cell biology is ultimately meant to deliver. Start with exercise. Fix the metabolic environment. Add the foods. Consider structured eating windows if you don't have contraindications. Get your vitamin D and omega-3 status checked. Sleep with intention. Those are not 7 steps to double your stem cells. They are 7 inputs to a biological system that is more intelligent than any supplement, more responsive than any injection, and more durable than any 30-day protocol, provided you give it the conditions it was designed to operate in. Synthesizing what we covered today, we looked at 7 studies showing that stem cell function across multiple tissue types is directly responsive to nutritional and metabolic inputs. The 5 mechanisms, the metabolic switch, autophagy activation, IGF-1 suppression, nitric oxide-mediated EPC mobilization, and W-anti-Notch pathway modulation, together explain a biological system that is not static, not fixed, and not independent of your choices. The protocol, organized by evidence strength, prioritizes exercise first, metabolic health second, specific foods third, structured fasting fourth with appropriate caution. The fact that you're spending time on this kind of information, looking for the actual science rather than the simplified headline, already puts you in a different category than most. Navigating health information in a landscape full of exaggerated claims is genuinely difficult. The research I walked you through today is the honest version. Some of it is definitive. Some of it is promising, but still evolving. I tried to tell you the difference. If this type of deep dive evidence review is what you come here for, the next video on cellular senescence, specifically the 7 foods shown in clinical research to reduce senescent cell burden in aging tissue, covers the complementary side of this biology. Stem cell activation and senescent cell clearance are 2 parts of the same regeneration story, and understanding both changes how you think about what to eat after 50. Leave a comment with your biggest takeaway or the question this raised for you. The questions you ask genuinely shape what I research next. Keep digging. I'll see you in the next one. Dr. Becker,